Dear viewers, this video is part 5 in the 6-part series on the cell and its functions in Medicine for Common Man series. Phagocytosis. Phagocytosis occurs in much the same way as pinocytosis, except that it involves large particles rather than molecules. Only certain cells have the capability of phagocytosis, notably tissue macrophages and some white blood cells. Phagocytosis is initiated when a particle such as a bacterium, dead cell, or tissue debris binds with receptors on the surface of the phagocyte. In the case of bacteria, each bacterium is usually already attached to a specific antibody. It is the antibody that attaches to the phagocyte receptors, dragging the bacterium along with it. This intermediation of antibodies is called opsonization. Phagocytosis occurs in the following steps. 1. The cell membrane receptors attach to the surface ligands of the particle. The edges of the membrane around the points of attachment evaginate outward within a fraction of a second to surround the entire particle. Then progressively, more and more membrane receptors attached to the particle ligands. All this occurs suddenly in a zipper-like manner to form a closed phagocytic vesicles. Actin and other contractile fibrils in the cytoplasm surround the phagocytic vesicle and contract around its outer edge, pushing the vesicle to the interior. The contractile proteins then pinch the stem of the vesicle so completely that the vesicle separates from the cell membrane, leaving the vesicle in the cell interior in the same way that pinocytotic vesicles are formed. Dear listeners, by the way, this educational video is made possible by PrescribeOn, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribeon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Lysosomes digest pinocytotic and phagocytic foreign substances inside the cell. Almost immediately after a pinocytotic or phagocytic vesicle appears inside a cell, one or more lysosomes become attached to the vesicle and empty their acid hydrolysis to the inside of the vesicle. Thus, a digestive vesicle is formed inside the cell cytoplasm in which the vesicular hydrolysis begin hydrolyzing the proteins, carbohydrates, lipids, and other substances in this vesicle. The products of digestion are small molecules of substances such as amino acids, glucose, and phosphates that can diffuse through the membrane of the vesicle into the cytoplasm. What is left of the digestive vesicle is called the residual body, represents indigestible substances. In most cases, the residual body is finally excreted through the cell membrane by a process called exocytosis, which is essentially the opposite of endocytosis. Thus, the pinocytotic and phagocytic vesicles containing lysosomes can be called the digestive organs of the cells. Lysosomes and regression of tissues and autolysis of damaged cells. Tissues of the body often regress to a smaller size. For example, this regression occurs in the uterus after pregnancy, in muscles during long periods of inactivity, and in mammary glands at the end of lactation. Lysosomes are responsible for much of this regression. Another special role of the lysosomes is the removal of damaged cells or damaged portions of cells from tissues. Damage to the cell caused by heat, cold, trauma, chemicals, or any other factor induces lysosomes to rupture. The released hydrolysis immediately begin to digest the surrounding organic substances. If the damage is slight, only a portion of the cell is removed and the cell is then repaired. If the damage is severe, the entire cell is digested a process called autolysis. In this way, the cell is completely removed and a new cell of the same type is formed, ordinarily by mitotic reproduction of an adjacent cell to take the place of the old one. The lysosomes also contain bactericidal agents that can kill phagocytized bacteria before they can cause cellular damage. These agents include the following: 1. Lysozyme, which dissolves the bacterial cell wall; 2. Lysoferrin, which binds iron and other substances before they can promote bacterial growth; and 3. Acid at a pH of about 5, which activates the hydrolysis and inactivates bacterial metabolic systems. Autophagy and recycling of cell organelles. Lysosomes play a key role in the process of autophagy, which literally means to eat oneself. Autophagy is a housekeeping process whereby obsolete organelles and large protein aggregates are degraded and recycled. Worn-out cell organelles are transferred to lysosomes by double membrane structures called autophagosomes, which are formed in the cytosol. Invagination of the lysosomal membrane and the formation of vesicles provides another way for cytosolic structures to be transported into the lumen of lysosomes. Once inside the lysosomes, the organelles are digested and the nutrients are reused by the cell. Autophagy contributes to the routine turnover of cytoplasmic components. It's a key mechanism for tissue development, cell survival when nutrients are scarce, and maintenance of homeostasis. In liver cells, for example, the average mitochondrion normally has a lifespan of only about 10 days before it is destroyed. Synthesis of cellular structures by endoplasmic reticulum and Golgi apparatus. Endoplasmic reticulum functions. The extensiveness of the endoplasmic reticulum and Golgi apparatus in secretory cells has already been emphasized. These structures are formed primarily of lipid bilayer membranes, similar to the cell membrane, and their walls are loaded with protein enzymes that catalyze the synthesis of many substances required by the cell. Most synthesis begins in the endoplasmic reticulum. The products formed there are then passed on to the Golgi apparatus, where they are further processed before being released into the cytoplasm. First, specific products are synthesized in the specific portions of the endoplasmic reticulum and Golgi apparatus. Protein synthesis by the rough endoplasmic reticulum. The rough endoplasmic reticulum is characterized by large numbers of ribosomes attached to the outer surfaces of the endoplasmic reticulum membrane. Protein molecules are synthesized within the structures of the ribosomes. The ribosomes extrude some of the synthesized protein molecules directly into the cytosol, but they also extrude many more through the wall of the endoplasmic reticulum to the interior of the endoplasmic vesicles and tubules into the endoplasmic matrix. Lipid synthesis by the smooth endoplasmic reticulum. The endoplasmic reticulum also synthesizes lipids, especially phospholipids and cholesterol. These lipids are rapidly incorporated into the lipid bilayer of the endoplasmic reticulum, thus causing the endoplasmic reticulum to grow more extensive. This process occurs mainly in the smooth portion of the endoplasmic reticulum. To keep the endoplasmic reticulum from growing beyond the needs of the cell, small vesicles called endoplasmic reticulum vesicles or transport vesicles continuously break away from the smooth reticulum. Most of these vesicles then migrate rapidly to the Golgi apparatus. Other functions of the endoplasmic reticulum. Other significant functions of the endoplasmic reticulum, especially the smooth reticulum, include the following. 1. It provides the enzymes that control glycogen breakdown when glycogen is to be used for energy. 2. It provides a vast number of enzymes that are capable of detoxifying substances such as drugs that might damage the cell. It achieves detoxification by processes such as coagulation, oxidation, hydrolysis, and conjugation with glycuronic acid. Golgi apparatus functions. Synthetic functions of the Golgi apparatus. Although a major function of the Golgi apparatus is to provide additional processing of substances already formed in the endoplasmic reticulum, it can also synthesize certain carbohydrates that cannot be formed in the endoplasmic reticulum. This is especially true for the formation of large saccharide polymers found with small amounts of protein. Important examples include hyaluronic acid and chondroitin sulfate. A few of the many functions of hyaluronic acid and chondroitin sulfate in the body are as follows. They are the major components of proteoglycans secreted in mucus and other glandular secretions. 2. They are the major components of the ground substance or non-fibrous components of the extracellular matrix outside these cells in the interstitial spaces which act as fillers between collagen fibers and cells. 3. They are the principal components of the organic matrix in both cartilage and bone. And 4. They are important in many cell activities, including migration and proliferation. Processing of endoplasmic secretions by the Golgi apparatus. Formation of vesicles. As substances are formed in the endoplasmic reticulum, especially proteins, they are transported through the tubules toward portions of the smooth endoplasmic reticulum that lie nearest to the Golgi apparatus. At this point, transport vesicles composed of small envelopes of smooth endoplasmic reticulum continually break away and diffuse to the deepest layer of the Golgi apparatus. Inside these vesicles are synthesized proteins and other products from the endoplasmic reticulum. The transport vesicles instantly fuse with the Golgi apparatus and empty their contained substances into the vesicular spaces of the Golgi apparatus. Here, additional carbohydrate moieties are added to the secretions. Also, an important function of the Golgi apparatus is to compact the endoplasmic reticular secretions into highly concentrated packets. As the secretions pass toward the outermost layers of the Golgi apparatus, the compaction and processing proceed. Finally, both small and large vesicles continually break away from the Golgi apparatus, carrying with them the compacted secretory substances and diffusing throughout the cell. The following example provides an idea of the timing of these processes. When a glandular cell is bathed in amino acids, newly formed protein molecules can be detected in the granular endoplasmic reticulum within 3 to 5 minutes. Within 20 minutes, newly formed proteins are already present in the Golgi apparatus, and within 1 to 2 hours, the proteins are secreted from the surface of the cell. Dear viewers, this is the end of part 5 in the 6-part series on the cell and its functions in Medicine for Common Man series. Dear viewers, thank you for watching the video.